Thank you very much, sir. Though you said it's brief, I felt it a little bit long, actually.
No, sir.
Anyhow,
Okay.
thanks for your wonderful introduction, sir. I'll take up this session. I'm very happy that I got an opportunity to have my discussion on the National Science Day. First of all, I would like to wish all the participants a happy National Science Day. Today we are celebrating. Hope my voice is clear.
Yes, sir. Clear, sir.
Yes, sir. Clear, sir.
Yes, sir.
Thank you. And slides are visible.
Yes, sir.
Yes, sir.
So
Yes, sir. Yes, sir. Visible,
my
sir.
first slide. Okay. Thank you. Thank you very much. Well, today I have a plan to take one of the most general aspect, probiotics. Probably you all are aware of it. And what exactly we are doing in our laboratory related to probiotics that I wanted to share in this 1 hour, 15 minutes of session. I have been asked to give a time for discussion, more time for discussion. So I'll be very happy if you can ask me any queries during my presentation, or else at the last, if you can ask me any of your interested queries, I'll be very happy to interact with you. So let me take my topic, probiotics, current status and future prospects. So I would like to say the word probiotics is one of the buzzing word nowadays. Like you all are very familiar with this word, probiotic. So initially, the word probiotic, we thought that it is something related to food. It is something being consumed along with the food. Like initially, even though there are around 33 participants, I'm damn sure whenever I said the word probiotics, the one thing which comes in your mind is curd, which is most familiar fermented food, which is widely being consumed. Most of the people think of curd whenever we said probiotic, and of course, it is right. It has started from curd. Then after, slowly, it has spread in different arena. Like I have some pictures here, which are exclusively used in the food. So from curd, it has moved on to the fruit juice. You can see here, there are fruit juices, different fruit juices supplemented with probiotics, and they named it as probiotic fruit juices. Then after, the curd has been modified into a different flavors, made it as flavored probiotic curds. And one of the most interesting commercialized, which is being available throughout the world, which has come from the Japan, that is Yakult, a probiotic drink. Selectively, the probiotics have been incorporated and made it available to all the persons who consumes this. And of course, if you go to any supermarkets, you will find Yakult in India and as well as in our local places like Mysore, Bangalore. And from there onwards, the probiotics have entered into the chocolates. You can see our Amul, which has come out with probiotic chocolates. And not just Amul, and there are many other companies also, they have come out with probiotic chocolates. And people started realizing the importance. They started asking for some more, that there are specialized probiotic tonics have come. You can see here, different tonics where it is being given as a medicinal supplement. I'm very happy to see that some of you who likes beer, there are beer with probiotics. So you can see a fermented beer having supplemented probiotics. And to my surprise, when I went through that, there is a water which is available as probiotic water. It is really a surprise for me. A drinking water is supplemented with probiotic, and I don't know how people are interested in drinking probiotics. Then initially we thought that it is only restricted to food. But then after, it has entered into the medicine, because realizing the importance of probiotics, people started thinking about medicinal aspects. People started telling about its importance in medicine. There are so many different probiotics which are given it as medicines. And you can see here, probiotic capsules, a immunobiotic powder to influence the immunity against different pathogenic bacteria associated with diarrhea. And today in market, there is one sachet which is being given to a children having different probiotics, a combination of probiotic, a consortia of probiotic. And to my surprise, in this faster world, 3 different capsules. Somalingappa, can you please unmute your... Okay, thank you. To my surprise, 3 different probiotic-based medicines, that moodobiotic, a mood biotic to enhance your mood and to reduce the stress. People are under stress. And of course, to reduce your weight, Probio Slim, they are really surprising for me. So from fruit to medicine, I thought probiotics may also be used in medicines. But later onwards, when I see some more probiotic products, I really started thinking, where are we going with the probiotics? You can see here, a probiotic moisture is there in the field of beauty, cosmetics. There is a probiotic lipstick is there. A probiotic skincare is there. A probiotic toothpaste is there. A probiotic deodorant is there. And probably without picture, if I would have shown, many of you may not believe at all this. How is it possible to have a deodorant, toothpaste, skincare, lipstick? Until and unless I see the product pictures and companies, even I did not believe. So people are thinking in a different ways. And to my surprise, a floor cleaner detergent is with the probiotic. Somebody who is interested in knowing how floor cleaner can have a probiotic, please, you can visit this website, Biotic. And moisturizer, hand cleaning solution. We all very well aware during COVID, it is with probiotic now. And you can see a toilet cleaner, a floor cleaner, a probiotic cleaning agent. And these probiotic aspects, when I see into the details, they have compared how good they are. My question was, whether can we use it? It is being used, it is being commercialized, and it is also being compared with other chemicals, how good these are. And probably if you're really interested in knowing how good these are, you can read this slide, wherein they say it is good in cleaning, it improves the ability, it enhances the flavor, and all those aspects has been given. How good is that? I was really thinking, where are we heading towards the probiotic actually? Is it an end for probiotic or is it a beginning for probiotics? That's why I thought current status and future prospects. With my curiosity, I looked into the probiotic market in world as well as in India. All the statistics says that in USA, in Europe, in Australia, and of course in Asia, there is a high market for probiotics. And whatever we see, maybe in another 5 years or in a decade, they say that there is an increase of 10 times the probiotic market. So one time only, I am seeing all these aspects, all these products. If there is a 10-time increase, I am really surprising where and all we see probiotics. It is not just the statistic given by the USA market. Currently, we have the market which is limited, like around $28 million USD. But it is expected to go up to 75 billion in future, the market size of all around. Is it only in USA and Europe? No, even in India also. This is the market for probiotics in India. It clearly says that there is a huge increase in probiotics. So this is the preamble or background what I would like to give. Where are we heading towards the probiotics? So what are these probiotics? The term probiotics has a definition. It is being derived to say any organism can be a probiotic or cannot be a probiotic. To say probiotic, there is a set of regulations or rules or criteria. So there are certain bacteria and of course yeast have these set of criteria to be claimed as probiotics. There is a big list of bacteria. And there is only one yeast which is being recognized as probiotics. That yeast is Saccharomyces boulardii. Of course, even though there is one yeast, the market is huge for that yeast. So it is competing with bacteria too. So on the whole, these bacteria and yeast, they are useful bacteria. They are friendly bacteria. Or they are friendly microbes as there is yeast. Most of these probiotic organisms are microorganisms found in food, different fermented foods, different natural foods and products. And also, they are known to be present in the gut of animals, including humans. And of course, flies. There are reports which are being isolated and characterized from different sources of flies, worms, animals, and of course, human beings. So to define these probiotics, a live beneficial microbes, which when administered in an adequate amount exerts health benefits to the host. So this is the definition being given by the WHO.
मग ती सांगा लागले सगळ्यांचा केला माझा केला नाही मी काल आलो नव्हतो बरं
Shashilekha Patil, please unmute. And which is being given as a GRAS status, generally regarded as safe. the whole probiotic organism which are being considered to use, they comes under GRAS status. Bacteria and microorganisms attaining GRAS status, it is a feather to microorganisms. And of course, since it is considered as GRAS status, so one question which comes in our mind, is it safety assessment is required or not? Most of the time it is not required. But nowadays, they say that to be on safer side, since it is going to involve or interact with many other bacteria, better to know their antibiotic susceptibility pattern, or antibiotic resistance carrying genes are there in probiotics or not, because they are presented in the arena of microorganisms. Well, these probiotics, for the first time, observed by the L.E. Mechnikov, probably you all know, during the early 1900s, in the yogurt, and he said that yogurt is good for health. Then next, after almost around 60 years, again, they rediscovered the word which are being, the sentence which is made by the L.E. Mechnikov, by Lilly and Stitwell. They coined the good for health comes from the bacteria which are present in that. They termed as probiotic. Initially, the bacteria, among a group of bacteria, the lactobacillus was the major probiotic organism, which is being introduced as first probiotic species, Lactobacillus acidophilus. Then after, Bifidobacterium. So Lactobacillus and Bifidobacterium are well-known probiotic cultures. Then there is a big list which comes in that. So as on today, we have more than 10 Lactobacillus species, around 7 Bifidobacteria species. Is it only Lactobacilli and Bifidobacteria? And of course, other organisms can also have the set of probiotic characteristics and to be named them as probiotics. Like there is a list here: Bacillus, Enterococcus, and probably some of the Enterococcus species being named as pathogens. But some of the species of Enterococcus species are named as probiotics, good organism. Okay, the list goes on. And the list is being still increased. It is expanding. There are many more organisms being listed into this. So where do they come from? Most of the probiotic organisms so far being isolated, so far being characterized, the majority comes from the food, what we consume. And I have a slide slightly related to India. So there are many there are many traditional foods have been documented throughout the India. You can see some of them here in this picture being characterized, isolated probiotics from these products. So what I would like to say, the Indian foods, most of the Indian fermented foods are rich in probiotics. Is it only available in the foods? as I said initially, they are also associated with human gut. Until 1990, people never thought that there is an association of probiotic organisms in the gut. But the scientists have studied that there are more than 1,000 species, 7,000 strains are associated in gut microbiota. Whether all are probiotics? Absolutely no. But the majority ones are the probiotic organisms. So when we see the diversity in the GI tract or a gut of human beings, in stomach, in small intestine, and in large intestine, where you can see there is a huge association of microbial diversity. Since the pH varies in these three conditions, and of course, since the food being eaten passed through these, maybe a stomach, maybe a small intestine, maybe a large intestine, there are many. And among the bacteria which are present in these regions, some of the common probiotic organisms being isolated, characterized, reported, are known to be present in these regions. Some of them are present in all the three regions. Some of them are present only to a specific region. That means they are exerting a beneficial aspect as a common or a normal bacteria in the gut region. So whenever we isolate these organisms, either from fermented foods or from the different hosts, how do we say they are probiotic strains? The Probiotic Association, the World Probiotic Association, along with WHO, has come out a set of criteria to say that this organism or this strain is said to be probiotic. The major 7 criteria to say they are probiotic, first of all, we should isolate them, identify completely up to the gene level for the identification. Then make sure that they are harmless. And whenever we give any probiotic strain, it should have the ability to go and adhere in the GI tract, in the any host. And since there is a huge pH variation, the organism should be resistant to acidic condition when it enters into the stomach. If the same organism is known to colonize either in small intestine or large intestine, they are supposed to resist to or tolerate the bile juice. If it is in the gut region, the large intestine, it should also have the ability to colonize the epithelial cells, where epithelial cells are depending on microorganism for their energy, because most of the undigested food is being digested or utilized by the organisms which are present in the gut region. So when it comes to safety assessment, the antibiotic susceptibility is one of the important things which we are supposed to test. Nowadays, it is highly being recommended. And then we can conclude that bacteria is so-and-so as probiotic. So whenever a probiotic is being given, how exactly they work? How can they exert the beneficial aspect? This is one possible diagram which tells that this organism may be used with the different aspects which can compete with other pathogens which enter into our body. Like there are three methods which are being discussed. One is computation for nutritional sources. Whenever we take probiotics, if at all, if any pathogens are there, they compete for the food sources. And the second one, they have the ability to produce different antimicrobial substances during different phases of their growth. And the third thing is, they compete with the pathogens to adhere into the epithelial cells. And you can see in these pictures, it is clearly being shown in the control group where there is no food is being given, any nutrients are being given. Consider this red ones are pathogens. If there is no probiotics, these pathogens gets these nutrients. If there is a probiotic group, these probiotics competes with these nutrients so that there is a lesser chance of getting nutrients for the pathogens. In a second way, there are many probiotic organisms have known to produce antimicrobial substances, different antimicrobial substances, which are generally termed as OSINs, like bacteriocins and other many compounds. And these antimicrobial substances, they go and interact with the pathogenic bacteria. And there is a possibility of affecting those pathogenic bacteria. And the third thing is computation for adhesions in two ways. One is whenever probiotics are there, they initially can go and bind onto the epithelial cell. They don't allow the pathogens to go and bind onto the epithelial cells, gut epithelial cells or other cells. And one more aspect which is being studied is co-aggregation. Means they just go and interact with the pathogens and it forms an aggregation, like they bind them so that they don't allow bacteria to bind onto the pathogenic bacteria to bind onto the epithelial cell. This is how they exert a beneficial aspects against different pathogenic bacteria. So by looking into the importance, the probiotics have been widely used for their medicinal aspects, for their health aspects. And you can see here, the lactobacillus species, Acidophilus, which is being used to regenerate the skin region. A bifidobacteria is used to prevent the obesity. A Streptococcus thermophilus organism used to improve the nutritional status. So list goes something like this. And you can see in this picture wherein the different medicinal value of the probiotics have been taken out, which lowers the blood cholesterol, which acts as immunomodulators, which is one of the better treatment for those who have lactose intolerance. And this is also being used for the prevention of some of the cancers, which is being researched nowadays. There are articles, the probiotics against cancer. I have very little knowledge into that. And to prevent diarrhea, and to produce some of the vitamins and growth hormones, And not just into this, there are many of different aspects. They are not just as a medicinal aspects, and of course, the organisms nowadays being used in the industries as well. Earlier, we used to use a normal bacteria in industries, but nowadays, industries are thinking to use a probiotic organism for some of the dairy product preparation, for some of the food related aspects, and for some of the fermented food production. And here are some examples. So for bread manufacturing, we all know yeast was used, a baker's yeast, but people started looking for a probiotic yeast to prepare the bread. And of course, the dairy products like cheese, yogurt, and many dairy products, people started using probiotic bacteria. Some of the meat processing industries, to tenderize the meat, they started using probiotic bacteria. In milk, they started adding as supplements. And for fermentation purposes, they started using a probiotic yeast. For biopreservation of some of the foods, they started using food biopreservatives. So it has entered into the production arena as a starter culture. So today, in order to say from the organism to say to the end product something like probiotic food, the Probiotic Association of India, World Probiotic Association, along with WHO, has come out with a set of studies to be conducted from isolating the organism till to produce the food. Something like identification, something like safety assessment, then after functional characterization, then after the efficacy, and of course, the labeling. It is must to say, oh, this is so-and-so is probiotic food. So those who are really interested to begin to isolate a probiotic organism till to the probiotic food, they can follow this set of experiments. Just to highlight some of the important experiments which are needed, like initially you need to isolate organism from different sources, identify that organism, species level. If possible, please deposit that organism in the safety depositories. Then after you conduct the in vitro experiments, there is a set of experiments which you need to conduct initially and to say this is probiotic organism. And if it founds excellence in its activities, then we can think of taking into the clinical trials. Since they comes under GRAS status, it is recommended to use for both animals as well as humans. It is recommended to use DBPC, that is double-blinded, randomized, placebo-controlled human trials can also be conducted. So very important thing, the placebo, one control group has to be maintained, because having a placebo clearly tells that the importance of probiotics, because many times whenever humans are being given that probiotics, before taking the product itself, they'll be thinking that, yes, I am eating something good. It exerts the beneficial effect. So that mind setup itself improves their health. So for this purpose only, it is compulsory to have a placebo effect. Then go for functional characterization like in vitro states and animal test, other aspects. Then we can see the effectiveness. Then after it can be controlled, it can be converted into a product. Then what is the bioavailability when we give it in a product as a starter culture, as a freeze-dried powder, as a liquid, it is being supplemented. Well, what am I doing in my laboratory related to this probiotics? In my laboratory, along with my research scholars, I am trying to isolate probiotic organism from different probiotic products, substrates, and I am trying to isolate both bacteria as well as yeast. Since I came from the mycology background, I worked on mycotoxins. I was thinking, is there any better method to modulate or to control these mycotoxins and mycotoxigenic fungi by using probiotic organisms? Because for my PhD, I worked on the cereals and I got to know that there is a problem of mycotoxinic fungi and mycotoxins in cereals, especially in India. Then I thought of controlling these fungi and mycotoxins. That is how my research has initiated. Well, in my laboratory, we have collected different traditional fermented foods in and around Mysore, in and around Karnataka. And of course, some of the products have also come from the different states like Jharkhand, Himachal Pradesh. So we have collected those fermented products, and probably you can see some of the fermented products are very well known to you. So one of the important product which we published, which is highly recognized among my publication, is a probiotic isolation from the Nira. Probably you all know what is Nira. It's a fermented drink directly taken from a coconut tree, which is being consumed in and around Mysore and of course other parts of the Karnataka as well. And there are different other products. So far, we have isolated around 787 different isolates from different food products. Whether all bacteria being isolated from the products or foods can be termed as probiotics? Absolutely no. In the food and products, along with probiotic organism, there are other bacteria too. Not all bacteria are probiotic organism. Then we need to do preliminary characterization. From 787, we came back something like 251 during our preliminary studies. Is it preliminary studies exactly tells that they are probiotic organism? Again, absolutely no. From 258, we have come out something like 124. And these 124 again studied for set of probiotic characteristics and found to be probiotic organisms as they have shown some of the significant results to be claimed them as probiotic organisms. And you can see some of the strains which are being maintained in our laboratory. Currently in our lab, as a library, we are maintaining We can claim them as probiotics, but we really don't know what for they can be used. But our primary target is these probiotic organisms can we use against mycotoxinic fungi. This is what is all about. So this is one of the paper which is being published by using one organism, a probiotic organism, which is isolated from a probiotic, from a fermented food. The organism identified as Lactobacillus plantarum. Initially, we have targeted Aspergillus species. As you all know that Aspergillus is one of the major mycotoxigenic fungi throughout the world. And there are mycotoxins being produced by this Aspergillus species. And to our best of our knowledge, you all know aflatoxins, aflatoxins. Tippeswamy sir. Tippeswamy sir. Yeah, thank you. We started screening against Aspergillus species, different Aspergillus species. Like we have targeted Aspergillus flavus, Aspergillus acraceus, Aspergillus parasiticus, fumigatus, carbonarius, like all are known to be mycotoxinic fungi. So the initial screening, when we have seen the agar overlay method was being performed, and we found some of the isolates have the ability to control these organisms. Up to 3 to 4 days, because the fungi needs to be grown up to 7 days. Up to 3 to 4 days, the inhibition efficacy was excellent. But after 5 days, slowly the fungi start dominating and the spores started spreading. But among all the isolates we have screened, we got one best organism which can control many Aspergillus species put together. That organism is being taken for further studies. Here you can see its ability to inhibit the different Aspergillus species. And the result, inhibition preliminary result is rechecked again with agar spot assay. And we found that this organism, Lactobacillus plantarum, shown to be effective against Aspergillus acraceus among all the different Aspergillus species we have targeted. Is it sufficient if it can inhibit only up to the mycelia of a fungi? Of course not. The fungi needs to be targeted at different sites. So when we looked into the efficacy, after 5 days, the conidia of a fungi started dominating the inhibitory effects. Then we started screening the organism as well as the metabolites produced by the probiotic organism for their efficacy to control or to inhibit the conidial germination. If conidia is being spread, if it is being germinated, then only it can spread as a colony. So that's why we have tested the organism and its metabolites to inhibit the conidial germination. And here you can see the conidia is being inhibited. The conidial germination is being inhibited. So they are not allowing the conidia to germinate. This tells that so fungi can be controlled. So when the Aspergillus is being controlled or targeted by the organism, so where and all it is acting upon on a fungi? This is our next question to be asked. So when we have done a scanning electron microscopic studies, and of course transmission electrons microscopic studies, we got to know that at different levels, the bacteria and its CFS or metabolites act upon fungi. So where and all it is being targeted? When we say fungi, the hyphae is important or a mycelia is important. So probiotic organisms are surrounded or attached over the hyphae of a fungi, and it is able to control or break or damage the hyphae. Our second best target was it able to control the spore production? Then what is its impact on a spore bearing structures or spore producing structures? So when we consider the Aspergillus species, the spore bearing structure is a vesicle, a conidiophore. So the probiotic organism, Sureka, Madam Sureka, thank you, ma'am. So probiotic organism and its CFS could damage the conidiophore and the vesicles. It is being shown here. So this is just to make sure that at different angles, the fungi is being targeted. And some of the studies being conducted at molecular level, we have tried the qPCR and RT-PCR to know the regulation of genes associated for ochratoxin production. It is found that the metabolites or probiotic organism is able to control the production of mycotoxin. So at different angles, we found that the organism is able to control the fungi. Then what next? Then we thought of applying these metabolites where there is a problem of this fungi. When we found that groundnuts or the peanuts are highly susceptible for this Aspergillus attack. There is high report to say that the contamination of Aspergillus in cereals and of course the oil seeds like groundnuts. So here you can see the biopreservative efficacy of the metabolites produced by the plant atom. We have tested against the peanuts and we got to know that this is the control group where the first plate artificially inoculated with Aspergillus flavus. All seeds are filled with the organism. And next plates are dipped into the CFS and they could completely control the growth of Aspergillus acraceus against the challenge inoculation. And one more study we targeted in poultry feeds. In all the poultry feeds, people are using toxin binders to control the toxin. And of course, it is very well preserved to avoid the fungal growth, because chicks and poultry are very sensitive to mycotoxins. So in poultry feed, also we have tested its efficacy, and we found that if any contamination is there, the probiotic CFS, which can act as a biopreservative, you can see here. And again, the same feed is being subjected for the total fungal biomass presence by targeting the ergosterol, one of the growth hormone of the fungi. Then after, the same thing which is being subjected for qPCR in order to know the copy number of a fungi. The same feed is also being tested for the production of mycotoxins, and we found that the organism inefficient to mitigate mycotoxins as well as the fungi. And this is being reported, published in one of the elsewhere journal, the Food Bioscience, and you can find that. Is it only Aspergillus species what we have targeted? Of course, we have also targeted different mycotoxinic fungi. So far, what all I have explained for Aspergillus acraceus, the concept, methodology remains the same. And again, we targeted one more mycotoxinic fungi. That mycotoxinic fungi is fumonisin producing fungi. The organism is Fusarium verticillidis. Previously, it was known to be Fusarium moniliforme, but now it is being named as Fusarium verticillidis. The organism was completely controlled, and the mycotoxin production, the fumonisin production, is completely controlled by one more organism. That organism is Lactobacillus brevis. So this is another a probiotic organism which we being characterized from different fermented foods, and we found that effectively it can control these mycotoxinic fungi. And this is also being published in one more journal, that is Frontiers in Microbiology. Should we target always the organism? Can't we target the metabolites? This was our question. So when we had a plan to know the answer for this, we found one organism, that Lactobacillus plantarum. The CFS of this organism, cell-free supernatant, found to be effective against one more Aspergillus species, that is Aspergillus parasiticus, which is second most aflatoxin producing organism. And probably you can think that why the first most significant producing Aspergillus species is not being targeted. And of course, we have done that. And that report I am not discussing, because that paper, we are working on that and we are yet to publish that work. So the CFS found to be effective against Aspergillus parasiticus. The set of experiment clearly conducted using the CFS of this organism has shown that it can completely control the aflatoxin production. And of course, it can also control the growth of Aspergillus parasiticus. Is it only mycotoxinic fungi is being studied at our lab? Recently, some of my students are also interested in working against some of the pathogens. So we have selected some of the pathogens like Salmonella, which is one of the major organism. as human pathogen. And of course, it is also one of the major problem in poultry also. This is the reason why we have selected Salmonella. And we found that, so one more strain of Lactobacillus plantarum, and of course the Enterococcus faecium, found to be effective against Salmonella. So for this, we need to develop different set of experiment than the fungi. So we have targeted at different levels, whether it can control the bacteria in the co-cultivation, whether it can control the biofilm formation by this organism, by the organism as well as CFS. It is found to be effective. And what is the MIC required for this organism? That is also being tested. It is found that the metabolites produced by the Lactobacillus plantarum can effectively control the Salmonella and Enterobacter faecium. And you can see here a different percent of active component of the CFS. And we found that between 10 to 15%, it is effective against both the organism. So what does it contain in this CFS? It is also being tested by doing a set of experiment, whether the protein is involved in this activity or organic acids are involved in this activity or something else. So when we have conducted this set of experiment to know the active component of the CFS, we found that the organic acids produced by the Lactobacillus plantarum found to be effective against Salmonella. And when we have analyzed which type of organic acids are known to be present, so some of the common organic acids which are known to be produced by these probiotic organisms being found in the LC-MS analysis. And is it only Salmonella is being targeted? And we are also targeting one more food pathogenic bacteria, the Listeria monocytogenes. And same set of experiment is being conducted. And we found that it is efficient against Listeria monocytogenes. And this paper is yet to published. And is it only bacteria what we are working in our lab? And of course, we are also trying to work out on yeast probiotics. And we have isolated many yeast probiotics from food sources. And they are being characterized. And I'm not going in detail of yeast probiotics. Sometime in future, if I get a chance, then I have a plan to take exclusively on yeast probiotics. And it is being tested against both fungal pathogens as well as bacterial pathogens. It is found effective. And some of the volatile organic acids or compounds being produced by yeast, they're also known to control the growth of it. So some of the organisms which are found effective, we try to know the whole genome sequencing of the same. And we have done the whole genome sequencing of these two organisms. And I'll be representing here one organism, the Levulactobacillus brevis strain, which was effective in controlling the Fusarium verticillidis, being submitted for the whole genome sequencing in order to know why this has antifungal attributes, which genes are responsible for antifungal attributes. So when we have done the whole genome sequencing of the organism, which is being published at NCBI, And we found that there is a set of genes which are predicted from the different platforms like PROCA and RAST annotation has resulted. There are genes are present as a functional genes in these Lactobacillus brevis like T3PKS, type 3 polyketide synthase, and other bacteriocin related genes being present in these organisms, which represents its antifungal activity. So with this, I am nearing to my time. Since I am told to give minimum 10 minutes of time to discussion, so I would like to conclude my session. Is it only probiotics is important as of today? Of course, no. People started thinking in a different aspects. From probiotics, people started thinking prebiotics. They are the compounds which exert the growth of probiotic organism, which influences the growth of probiotic organisms which are present in the host. So different prebiotics have been studied. Can we use probiotics and prebiotics together? Yes, of course. Whenever we take probiotics, it is recommended to take prebiotics also. And put together, it is being called symbiotics. So probiotics and a prebiotic, which is known to enhance its growth whenever we take to exert its beneficial aspects. Better to take it as symbiotics. And recently, the whole world is thinking of postbiotics. The postbiotics are the compounds produced by the probiotic organism or the other organism, so which are released, may exert the beneficial aspect. So probiotics, prebiotics, symbiotics, and postbiotics. These are the 4 biotics nowadays making a buzz around the world. So I would like to thank my research scholars. So what all I discussed so far, it is completely work by my research scholars. I have a set of research scholars who are working in my lab. And of course, research scholars may not work without funding agencies. I would like to thank all the funding agencies which are made me to discuss some of the work which is being conducted in my laboratory, the sessions like this. And of course, some of the international collaborators who have supported my studies, like Kansas State University, and of course, University of Ottawa and University of Arkansas, and some of my collaborators who are working with me to get some of the significant outputs. I thank them also. And finally, I would like to thank the participants who are present in this session. So I would like to tell you that it is so unfortunate that we are not in the physical mode, and you all must experience the Mysore Refresher Course Center. You all must experience the ambience which is there in this HRDC, because we all have experience. It is a wonderful place, but somehow it is online mode, and I don't know how effective the program is all about. Somehow I don't like the online presentation at all. It is something like speaking to a computer without having any expressions. So I'll be very happy to take, if at all, if any queries or questions related to my presentation. Hope you all agree with my presentation, like whatever I shared so far.
Sir, I have a question, sir.
Please, ma'am.
Sir, few research articles showed that the patients with the type 2 diabetes have a significantly lower number of bacteria when compared to healthy people. Sir, instead of medicines or drugs, can we use these probiotics which produces butyrate to treat type 2 diabetes, sir?
Yes, ma'am. I mean, they say that especially this lifestyle related diseases, mainly due to the reason of dysbiosis. Dysbiosis means we are going to lose the common commensal in our gut region. So they are making a huge imbalance in the dietary pattern. So that is leading to many lifestyle diseases. One among that lifestyle disease and food pattern is the diabetes. So there are many probiotics and postbiotics are being evaluated and found effective, and people are trying to restore these. And probably one of the thing which I would like to tell you is the WHO has recently accepted and released FMT, that is fecal matter transplantation, against some of the lifestyle diseases. It is already in the market. So I think there are many articles which are moving towards the diabetic. It can be done.
Thank you, sir.
Hello, sir.
Yes, sir.
Myself is Dr. Venkatesh, Danagari University.
Yes, sir. Yes, sir.
Nowadays, the doctors are suggested to take a probiotics for children. So,
Yes.
whether that is an good or not, whether that is very acceptable or non-acceptable, sir. Whether they, how long that will be helpful to retain their that probiotics in their, especially the kids. And is there any adverse effect if you are taking externally in that bottle license probiotics that are available in the commercial, for especially for the kid, for the growth, and any effect there in their digestive tract?
Sir Venkatesh, sir.
Hello, sir.
It is highly recommended to take probiotic consortia for the diarrhea. And as far as my knowledge is concerned, doctors do not prescribe any antibiotics for children if they have the problem of diarrhea nowadays. They are giving one sachet, which is called Econam, having a consortia of probiotics, 2 bacteria, and 1 Saccharomyces boulardii. Without giving any medication, directly they are recommending, or they are giving the probiotic itself. As far as my knowledge, there is no problem at all, and it is highly approved and accepted. Most of the doctors do prescribe only probiotics rather than the chemical treatment. No adverse effect found so far.
Hello, sir. Again, sir. Is there any purity checking when we are in the lab? Is it possible to check that whether that probiotics are there in sachet or not? How we can do if any minor protocol is available, sir?
Yes. So we do that also. The moment when any commercial product comes out, in order to exert any commercial product, their health benefits, that bacteria should be viable first.
Yes, sir.
Means the moment we take, it should grow in our gut. That means it should be active.
Yes, sir.
So we have brought many commercial capsules.
Yes, sir.
Grown on the particular media, and we are using them as our positive controls.
Yes, sir.
For our study. We have isolated Saccharomyces boulardii from the Econam sachet itself.
Okay. Right.
So that we are using it as a positive control. We don't say it is not good. We say along with this, it is good.
Okay.
So you can do that. They are all viable, sir, most of them, because they say this many CFU is bioavailability. You can see that. So when you culture that, they will grow.
Yes, sir.
Okay.
Sir, good afternoon, sir.
Good afternoon, Prof. Shankarji.
Good afternoon, sir.
Yes, sir. Prabhu, sir.
Sir, myself, Dr. Prabhu from Davangere City, sir, Biotechnology Department.
Yes, sir. Yes, sir. I know.
Okay, sir. Sir, one of our student isolated the lactobacillus from sheep milk, sir.
Okay.
MRS media they are used, sir. But when we gone for sequencing studies, sir, but it is amplified with 18S, that is hitting to the yeast species, sir, that is Pichia species.
Okay.
So what would be the next, sir, guidance suggestions from your side, sir?
It's good, actually.
That is sir. That can be considered further, sir, further studies.
It can be considered for further. When you have done in vitro studies, a set of experiments,
Yes,
if it is showing
yes,
some of the significant result, then you can go for the further aspects. And isolating yeast is highly recommended because we are well short of yeast probiotics.
Okay.
Only
sir,
one is there in the market.
Okay. Sir, one more thing, sir. Can we continue with the MRS media only, sir, for that species?
No, it is yeast, better to go for Zapadox, better to go for SDA, better to go for yeast mannitol extract agar. Like there are different set of media is available.
Okay, sir.
So better to go for yeast related and confirm that as yeast. Don't just go for 18S RNA. There are set of primers which are specific to yeast. Either Saccharomyces specific primers are there, or Pichia specific are there. Like these are the different yeast probiotics which we have isolated. And there are being Chloromyces and other aspects, and you can try that and you can isolate and identify them.
Okay, sir. Just it is preliminary work, sir, that it is hitting confirmed with the PCA, sir. That's all.
Sure.
Okay, sir. Thank you, sir.
You can recheck that. Otherwise, in goat milk, there are many other bacteria also might be there, and you can also try them because that is not the only isolate. Whenever we isolate from a natural source, we get many different colonies. So you can also try other colonies.
Okay, sir. Definitely, sir. Thank you. You tried with sheep milk, sir, from Raichur district.
Okay. You can try gram staining, and you see whether there are bacteria. Then you can also look for bacteria in that.
Yes, sir. We NaCl tolerance, bile salt resistance, pH resistance, all aggregation, co-aggregation, hydrophobicity, all done, sir.
Yes, sir.
After that we send for. That's all.
Preliminary characterization is done.
Yes, sir. Then we send for.
Hello, sir.
Yes, ma'am. Purnima, ma'am.
Hello, sir.
Yes, sir.
Yes,
thank you. It was a wonderful talk.
Oh, thank you.
You explained in a simple manner, sir. Everybody could understand.
Okay.
Sir, I have one simple question, sir.
Please,
instead of buying those sachets, sir, which are available in the market, can we use simple buttermilk which is available at home, sir?
Ma'am, you can use, but we are
buttermilk,
not sure.
Sir?
Yeah, we can use.
Yes, sir.
But we are not sure whether that buttermilk is rich of those selective isolates. Because when he is already under problem, better to recommend in required quantity. That's what WHO say. Whenever we give adequate amount of probiotic, then only it exert the beneficial effects. So in buttermilk, we don't know whether they have adequate amount because it's a concern. There are many organisms to be present.
Yes.
Some organism may dominate over these organisms. So that's why people are started isolating and then giving it in an adequate amount. And I don't completely neglect your answer because traditionally, our traditional knowledge just said whenever we have problems like that, better you take this juice, better you take this product, better you take this extract. All these may act as prebiotics for selectively.
Yes.
And then the buttermilk also known to contain some of the postbiotics, which are efficient nowadays. There are postbiotics taken from buttermilk.
Yes, sir.
Traditionally, we all know that during summer, better you take buttermilk. It keeps you cool. It maintains your health. It gives all those things. And nowadays, people are telling that maybe postbiotics in that buttermilk because we are going to churn. We are going to take only the liquid part of the curd.
Yes, sir.
So more of postbiotic, less of probiotics.
Yeah. Thank you, sir.
Go, ma'am. Thank you.
Hello, sir.
Yes, ma'am.
Sir, this is Navya from Davangere University.
Yes, most of them are from Davangere University. Please go ahead.
I completed my PhD from biotechnology.
Yes, ma'am.
Sir, after a long time, I'm happy to hear the mycotoxin related topic.
Oh, that's great. Okay.
Sir, my question is that irrespective of the lactic acid bacterial source, all kinds of lactic acid bacteria are effective against the mycotoxins or not?
No.
What are the major sources are effective against the mycotoxins?
That's what, ma'am. In our laboratory, we are maintaining around 251. As on today, we are maintaining around 251 uncharacterized, simply preliminarily identified probiotic organisms are there. So whenever we are interested in any particular mycotoxin or mycotoxinic fungi, initially we screen all the library against this mycotoxinic fungi. So we found that if we screen around 80 plus, we might get one. We consider that as a significant one and take further.
So all lactic acid bacteria, the probiotic bacteria, are from the dairy sources, or whether they are also from the rhizosphere soil?
No, madam. Not all lactic acid bacteria, not all LAB are considered to be probiotic. So there are huge number of species are there. Yeah, there are huge number of species are there, but 20 plus species have been considered, recognize them as probiotics. And whenever you isolate any strain, even if you get well-known like Lactobacillus rhamnosus, one of the well-known probiotic organism which is used in medicines. So if you isolate Lactobacillus rhamnosus, straight away you can't say it is probiotic until and unless you prove under in vitro studies and as well as in vivo studies. They say it might be strain specific too. Okay, I think thank you, one and all. It's lunch time.
Thank you, sir. Thank you so much.
Okay, thank you. Thank you.
So I request one of the participants to propose vote of thanks to Professor Sreenivasa.
So when we were attending refresher courses well in advance, we used to plan who will be doing welcome and who will be doing vote of thanks.
So this is because being online, I think we are unable to recognize who has to propose.
I will do, sir.
Yes, madam.
Before the start of any session, we used to discuss among us. Okay, I'll do this, I'll do this, you do this. That is better to share among yourselves, because at the fag end, if you decide